Filip Ilievski, who we had here at the center. We had the fortunate opportunity to meet him at the center at our GCE workshop and conference last August. Filip is originally from the Republic of North Macedonia, where he earned his pharmacy degree and then did his master's thesis in Professor Manfred Ogris's lab at the University of Vienna. During his undergraduate studies, Filip was awarded research internships at institutions such as the University of Cambridge and Helmholtz-Zentrum in Germany, working on topics ranging from GPCR signaling to antimicrobial resistance. Since 2021, Filip has been pursuing his PhD at Uppsala University in Sweden, under the guidance of Professor Magnus Johansson, where he is supported by the Uppsala Antibiotic Center. His research focuses on integrating chemistry, molecular biology, and biophysical methods to develop a technology he calls FLORENCE, a universal labeling method for single molecule tracking using genetic code expansion, which I believe he'll talk about today. Filip's skill set encompasses GC development, genome engineering, fluorescence microscopy, as well as protein design. He plans to defend his thesis in 2025 and is currently exploring opportunities in both academia and industry with the long-term goal of mentoring and leading future generations of researchers. Thank you for joining us today, Filip. We are most excited to hear about your research. Please take it away.
Thank you so much, Rick, for the introduction. Are you able to see my slide? I
see your desktop background.
Okay. And how about now?
I see it now. Perfect.
Yeah. Okay, great. Thank you so much, Rick, for the introduction, and thanks for everyone in the center for this opportunity. I'm actually very humbled when I see the list of speakers in the past and upcoming to be here and to present the research that I was guided under in the Johansson lab here in Uppsala. So I will be talking about, in general, protein translation and why is it that important. It's the fundamental process, then put an accent on single molecule tracking, which is the major technology that the lab uses, then comment on how this transitions with the genetic code expansion, and then talk about FLORENCE and the parts of FLORENCE. That is the optimization of the bacterial chassis, the optimization of the protein expression vector, and finally the application for single molecule tracking. So protein translation can be looked into two ways. This is the usual textbook example that we are studied in, where we have a ribosome that is composed of two subunits that reads codons along an mRNA and does protein translation. In reality, however, this is much more complex, and all of this process is actually very highly orchestrated. And now let's take a look into all of these parts. So there is, of course, initiation with all the initiation factors that are primarily into bacteria cells. So the Johansson lab actually focuses on studies in bacterial cells, and this is what I'll be talking about. So in the initiation, you need all of these factors to coordinate and come together that promote subunit, the ribosomal subunit assembly. After the initiation has occurred, you have equally complex process of elongation, where we have additional elongation factors such as EFTU, EFP, EFG, elongation factor 4, and finally you have protein termination that is equally complex in that sense. And you can see that all of these factors, so for example, EFG plays roles both in elongation, but also in post-termination complex release and termination. And now we know that this is central to life. And as I said, it's a very highly synchronized process. And all of the numbers here that come in the time on how much does it take to translate an mRNA, how much does it take for one elongation cycle to happen. This all comes down from in vitro test tube studies, where a plethora of scientists have isolated individual components, mix them in a test tube, and by quench flow or stop flow, fast kinetics and ensemble methods have measured intricately each of these parts. However, reality in living cells is different, and really the central goal of the lab, where I'm finishing up with my thesis, is what is the kinetics of protein translation in vivo in living cells. The protein initiation kinetics has been recently deciphered by one of my colleagues in the lab, where he tracked basically all of these initiation factors and got numbers for living cells. The same was done by tracking ribosomal subunits, but what is not known is there are still certain debates about the elongation factors, that is elongation factor term unstable, which was for the first time reported by the Weisher lab, and finally EFG that no one has really characterized for now. So if we go back to the in vitro studies, how this is done is basically that each of these kinetics processes is approximated to a Michaelis-Menten equation. And if you plug in a whole bunch of first-order differential equations, you will come up with the numbers of turnover kcat and km that later on translate into times. And this was, again, pioneered in Uppsala, in the Ehrenberg lab, where my boss did his PhD, and he was able, in his work, to decipher the proofreading and the selection of tRNA. And recently, one of the postdocs that I'm closely collaborating with, Annalie Borg, during her work in her study, she was able to measure the rate of translocation. But this was all done in test tube reactions by having each of these individual components and tuning their concentrations and reflecting them to a Michaelis-Menten kinetics. So this is all fine, I mean, as I said, but in reality, cells are much more complex. You have much more players that are in, and all of these reactions, we want to make sure that we want to find out like how real they are. And this is done by single molecule tracking. Now, single molecule tracking is actually a technology that takes its inspiration from astronomy. So the way that astronomers would look at the sky and follow the stars, it is the same way that a microbiologist or biophysicist will look at molecules inside cells. And just to bring this up closer to the audience, by tracking the position of these three stars over here, and by marking the position of Venus, scientists were actually able to figure out when was Starry Night painted, and it was somewhere in the 1825s. And basically, if you take, that means if they were able to calculate the trajectories of the Aries constellation that is depicted in Starry Night and went back, they could, of course, they would to some extent get the right timing. And if you, I mean, analogous to this, this is basically how, for microscopists or biophysicists, cells with fluorescent labels would look. The principle of single molecule tracking comes in the fact that you have a molecule that is labeled with a fluorophore. Most historically, there were fluorescent proteins. We'll talk about that. And then it binds to another molecule that it interacts with. And this interaction has a certain changes its diffusion. So by this, by calculating a diffusion coefficient, one can identify different diffusible states of this molecule. Most of the time, what you would be expecting is a free floating state of the molecule, a bound state where the diffusion of the coefficient, of course, will change. And this bound state usually means an interacting state or something like that. Based on the hidden Markov modeling, which by whose maths I'm not going to go behind, one defines certain parameters, and that is occupancy, that is the percentage of the state population, the dwell time, and the time spent into that particular state. And based on the diffusion coefficient, occupancy, and dwell time, one can, by plugging this into a model which works like some sort of a black box from a tool maker perspective, you will get, you will characterize the kinetic profile of a given molecule. What is very important in single molecule tracking to get good data are actually the long trajectories. So you want to label with dyes that have high photon budget. Now, to translate that mathematics into biology, you need to do certain biological controls. That is, you need to do mutations in the binding site. So for example, if you get a binding mutant, you will see that you will shift most of your population of free molecules into a bound state. Or if you apply antibiotics that are promoting the dissociation, or you have a non-binding mutant, or if you increase the concentration of the labeled molecule, you will shift the equilibrium or to a population where you have freely diffusing molecules. And this was done very nicely in the lab with this groundbreaking paper where they labeled ribosomes with a halo tag in a normal MG1655. And they found that at any given time, these ribosomes spend 90% in their bound state, regardless whether you're labeling the 50S or the 30S subunit. And other labs, such as, for example, the Kapanidis lab that pioneered this back in the days, they labeled RNA polymerase with a red fluorescent protein or a photoactivable red fluorescent protein, and they were tracking individual molecules, and they were able to find the diffusion of RNA polymerase and localize this around the cell. So this is where, I mean, if you notice what I said fluorescent proteins, halo tags, and these are labels that are pretty big. This is where the GC comes in super resolution microscopy. So again, very briefly, you have a designer amino acid with a designer system that gets incorporated to the stop codon, and you get your protein. It's a label. This is fantastic because you do a minimal perturbation of the protein structure and function, and you're improving the spatial resolution, which means that you have a very small tag. Your localization error at any given time point will be very low. Of course, in normal bacterial cells, this historically has been suffering quite a lot by the presence of the release factor 1, up until the point where researchers started making this release factor 1 deficient strains increase the protein yield, and they were getting less of the truncated proteins, with at one point were shown to be pretty toxic. So what is the tools at hand? Now, this all comes down with click chemistry, and these are the most commonly used unnatural amino acids because of their commercial availability. Up to the point where in 2012, GC Center introduced these tetrazine amino acids. So really the arsenal now that we have for click chemistry labeling is quite big and is getting bigger and bigger. Again, the problem always for biologists is to have it commercially available. And the dyes are mostly in the tetrazine configuration, the legendary Weiss letter probe, the body pipette metal tetrazine, with the invention, with the synthesis of the JF549 dyes that have very high photon budget, that are very well cell tolerable and very well cell permeable and washable. And of course, there have been the tetrazine, the sci-fi dyes, but the sulfur sci-fi dyes, which are not super bacteria friendly because of their sulfur groups. They cannot really penetrate, and they're, even when they do, they're like super unwashable. And I'll talk about this later. Again, fluorescent proteins and other tags like SNAP and halo tag, although they can be easily fused to a protein of interest by simple PCR, and they have the advantage of tuning the dye's brightness, means that you can engineer basically your protein, even if you have a dye that's not bright enough, to get high enough of a brightness. And that's, of course, a very big advantage for single molecule tracking. However, it is a problem for delicate and dynamic proteins because they're very bulky. If you have a protein from 10, 20 kilodaltons and the smallest tag that you have is around 25 to 30 kilodaltons, most likely you might run into trouble. And another thing is that, for example, for FRET studies, they do lack the site-specific resolution. Let's say you have a protein that doesn't tolerate being labeled at the N-terminal tag or the C-terminal tag. The way to go here will be with GC and click chemistry. And I mean, as I said, the problem is applying them to dynamic and small delicate proteins. And this is what I was talking about. So here's, for example, from the Delis Liang lab, where you can have, no matter how bad of a fluorophore you have, if you just engineer the protein, as they show in this panel over here, you can actually increase the brightness. So that is one of the advantages of this protein dye conjugates. And this, for example, can be very specific for a given ligand. The JF549 series is great, and especially the halo ligand that has been quite used in the community simply because it has this fluorogenic effect. That means when it binds the halo tag, the halo ligand binds the halo protein, the fluorescence increases. For some applications, I mean, this is great. And this is maybe where the GCE kind of lacks and would require a further optimization. But what people have actually done with GCE for super resolution microscopy is only label receptors and outer membrane proteins. So for example, this is one of the previous speakers, the work of Ivana Nikic-Spiegel, who labeled the insulin receptor. This was done in the Lemke lab. She has also done this in primary neurons, but most of the time, like if you look into the literature for super resolution microscopy, it all goes into membrane receptors. The group of Markus Sauer, for example, they use quite a lot of the click chemistry, and they have also shown that they can label adhesion GPCRs. But again, what I want you to take the take-home message from here is that all of these guys are membrane bound. And if you have a dye that doesn't necessarily penetrate the cells, it will be very easily, when you put the label on outside of the membrane, to basically label it. Most recently, the Stefan Hell lab used the GCE to label Philopodia, and here they were actually able to improve the localization error from 2.1 to 1.4 nanometers, so almost double compared to, for example, antibodies. Now, for single molecule tracking, there are only 2 studies that employ GCE, and that is one started back in 2016 from a neighboring lab, Johan Elf's lab, which project I kind of overtook. And that is, they were trying to label LAC-I. And back then, they didn't have any dyes that are cell penetrable. So they ended up, unfortunately, this ended up being unsuccessful in trying to label intracellular proteins. However, I mean, as I mentioned before, all of these studies were able to label outer membrane proteins and actually do single molecule tracking on outer membrane proteins. So for example, here you can see for an intracellular protein like LAC-I, there's not much difference if you label it with an unnatural amino acid and if you just apply the dye by itself. Like, you can't really distinguish between false positives and real positives. However, it works very nicely if you apply it for a outer membrane protein. And the same trick has been used by Natalie Elias' lab, where she labeled the epidermal growth factor receptor with a BCNK, but again on the outside membrane. So and of course they have tracked this and got diffusion coefficient, but for single molecule tracking, there really hasn't been any study for intracellular proteins, and that's where we come in. So with Florence, we want to first of all do single molecule tracking in bacteria. We want to get an MG1655 like genotype and phenotype, which is the classical lab strain that we work, which means that we need to get close doubling, have same doubling times, same self-morphology, and not off-label targeting. And that's why here we are selecting chassis, and I'll talk about this in a moment. We want to have an orthogonal translation system that is very highly orthogonal to the natural translation machinery. That means it doesn't charge native tRNAs, or it doesn't do any near-cognate suppression. The whole system is non-toxic and it's highly efficient. And finally, we want to select dyes and unnatural amino acids, which are, of course, bright and they're easily washable, because in microscopy, you really want to get rid of the background. So the chassis that we were looking in was the C321 series that was started in the Church lab. At first sight, these strains are MAT-S, biotin AB, Tolsi, and GalK knockouts, and they cannot grow at 37 degrees because they have the lambda red cassette inserted. And their parent strain is an actual MG1655 strain, so we focused working on these. There is, of course, the B95 strain from the Sakamoto lab, for us, it's a no-go for physiological studies because it is an BL21-like phenotype and not an MG1655 phenotype and genotype. And it actually, in the paper, they have reported quite high doubling times of 80 to 90 minutes. And finally, there's something which is called C123, which is again a strain from the Church lab, where they have removed all the rare arginine codons. But again, the doubling time seems to be quite a problem. But we focused on the C321 series, and I will show later. So this is an example of the C321 M9 adapted over here. If you grow it at 37, you get quite a lot of colony heterogeneity. If you then replace or remove the lambda red system, restore the MAT-S, and put back the biotin operon, you see that these colonies, in terms of size and morphology, are similar to MG1655, which means that they can grow now at 37. And since most of the chemical reaction take time at 37, we're actually able to work with these. We took and fixed all of these church strains by doing this particular fixing, and we checked their doubling times in different media: LB, M9 1% glucose, and ridge-defined media 0.2% glucose, because these are our imaging media. And we decided to go for the C321M9 adapted because it showed most consistent doubling time and physiology compared to an MG1655. So this is how the curves look. In LB, basically, they're virtually, to some extent, indistinguishable. In glucose M9, again, MG performs much better. In glucose 0.4, again, MG performs much better, but then this optimized strain, we call it GRO1, doesn't work that well. And the GRO2 is basically a fixed M9 adapted, which outperforms its parent in all of these imaging media. Okay. And this is a video that actually I would show from quite recently, and that is that we actually did some additional fixes. Here, for example, you can see in a mother machine that these cells look very uniform and they double very evenly. However, if you put the M9 adapted into a mother machine, it shows some sort of growth defect. And this is simply because of the lambda red system that it has in. And basically what we did was we just recently put back the mutations that were found by the Ellington lab back in 2018. So this is pretty reproducible. Okay, the vectors that we were working with is the p-vol vector. However, in this case, we historically, for GCE, it had this M-Mazei translation synthetase. But recently, we took the Alvus synthetases from the Yokoyama lab that come in 2 versions and were actually specifically engineered for bulky amino acids. And then we took this reporter of a super cyan fluorescent protein, where we put this reporter at the N-terminus with a codon context linker. So this is the context that was shown to work kind of quite nicely. And we put this on a 1 plasmid system. So we put the IPTG induction, so this is IPTG inducible. We put it on the p-vol vector, and we just put it into 2 different places. And we were wondering what is going to happen. And as a control, we have this double plasmid. So in an MG1655, but with arabinose induction and the TCO star axial isomer, we basically see that at 1 millimolar, orthogonal translation system 2, that is an M-Alvus system, outperforms M-Mazei. And it actually surprisingly outperforms this double plasmid system by simply looking at the slopes. What we were looking for in this case was the fastest protein production. And if we just measure the slope at the normalized fluorescence, we would get this synthesase to work the best, just as reported in the Yokoyama paper. Now, if we apply this into the 2 different recoded strains, first of all, you will see that in our fixed strain, we do see some sort of incorporation. What is very nice is that in this strain, the synthetase actually responds to different concentrations of amino acids. And those different concentrations of amino acids do not have any negative effect. And that is the same in GRO2, the one that we are currently working with. Although GRO1 shows a bit higher unnatural amino acid incorporation, the fact that GRO2 shows faster amino acid incorporation is what we decided to go for. Then we just ran a simple gel to label the cells with a JF549 tetrazine on the one plasmid system vector. So this is the band in the fluorescence for the SCFP, and we don't really see any off-label, off-target labeling, at least at this point. And this is like the positive control of an AAA, where we don't have the unnatural amino acid. And you can see that in principle, like I showed before, natural translation seems to be much better than unnatural translation in these recoded strains. Okay, but then we ask the next question: which of these clickable amino acids are best tolerated in this chassis? And it turns out, again, TCO star, for the one for which was engineered, is tolerated the best, following by the BCN endo and exo, and very little tolerance for the CPK or for TCO4. And this was again done in MG1655, but the results are the same basically in the recoded strain in LB media. Okay, now the important thing is that the structure of the dyes and your click adduct is pretty important. And why am I saying this? The tetrazines are natural quenchers of this moiety over here. And if we compare these two dyes, the JF549 dye and the Heidelberg dye, we see that by moving the tetrazine closer to the fluorophore, we have a bigger quenching effect. Now, in all fairness to say, over here, if you look at this graph, the JF549 is inherently brighter than the Heidelberg dye, at least twice as bright, but the turn-on ratio is quite low, higher in the Heidelberg dye compared to the JF549 dye, which would mean that we will have the increase in signal-to-noise ratio. What you need to take into account is that once you form these adducts, depending on whether you're reacting this with TCO or BCN, you might actually lose the fluorescence because of the adduct that is being formed. Okay. So now we move into microscopy, and we, up to this point, had like a 15 hours labeling protocol where we would actually wash extensively, label, get the protein production, and then finally look it under the microscope. And this was pretty long because the protein production took a lot of time. But by tuning out a little bit of the expression vector, which you will most likely read in our preprint, you will see how that was done. So this is just a microscopy control where we have the background of the cells. So these are the cells without dye. If we just add now the dye, again, we can wash it out. If we add the unnatural amino acid, we see all of a sudden that our stop reporter glows. If we now add the dye, we're able to see quite a high concentration of dots. And if we have like a normal lysine and again add the unnatural amino acid, we don't see any unspecific binding. And then if we do single molecule tracking, here, let me just try to, we can see these dots of a supercyan fluorescent protein just like moving around. And what we see here is that they have very sporadic free states. Now, in all fairness to say, this is not like super bright, but we're still actually able for the first time to show that in a recoded strain without any off-target, we can show bright spots. Unfortunately, compared to the halo, they bleach very fast, but this is like really the best we can get as a proof of principle. Now, we took the LAC-Y, fused it to halo, and had a halo dye or a click dye, and decided to validate this in terms of numbers. So what we were looking at here are basically just comparisons between the fast and the slow state. So we see that regardless of how you label the dye or this lac-Y, you would be getting lac-Y in a fast state. I mean, the diffusion coefficients are somewhat similar depending on the different methods, and the occupancies are maybe a little bit different. But this for us is showing that we can actually, with two labels, label one protein in living cells. Okay, so that's pretty much it. I will just finish with something that is like an improvement, because throughout my PhD, I have been iterating this quite a lot. And now we see that a small trick that if you remove the arabinose operon and make ARA E constitutively expressed, you can actually get higher protein yield based on the gel over here. So this is actually protein expression in 2 to 3 hours after induction. And I can, of course, discuss this after I'm done with the presentation. So to summarize, we have a recoded strain that outperforms the parental strains with physiology, which is like near MG1655. By proper induction of the synthetase, we can decrease the production time. There is no fitness caused by this M-alpha synthetase, and the dye photophysical properties are actually dependent on the unnatural amino acids. So depending on which click partner you use, you would get different brightness. We can use GCE for single molecule tracking. One of the disadvantages over here is that we have delicate expression control. That means that we have because this is done all on a plasmid, we get quite a lot of plasmid heterogeneity. And as Rick said, I had the privilege to be part of the GCE workshop, and one of my projects is a biosynthesis of click amino acids so that one can actually circumvent this inborn plasmid heterogeneity. And one of the libraries that we got from is the hit from the GCE workshop, which was actually quite great. I mean, I met great people. It was very educative. We actually now have what we believe is a very good synthesis for this click amino acid that has been so far the bottleneck in this whole project. I would like to acknowledge at the end my supervisor, Magnus, for his guidance, Annalie, who's been helping me quite a lot with another project on the translocation kinetics, Ivan, who's been instrumental in helping with the biophysics part, Linnea, who worked as a summer student on optimizing the protocol for the labeling, Clara and Ella, who started actually this protocol, this project in the Johansson lab, and Frida, who was working on the biosynthesis project. Garrett, my co-supervisor, who helped me with fixing the church strains, and everyone in the ELF lab, Jimmy Larson, who helped me with the microfluidics, and Vinut and Beer, who have been quite helpful in the plasmid construction. And finally, I would like to thank the GC for All Bulletin Board, and I have to, of course, acknowledge the funding from the Uppsala Antibiotic Center. And with that, I'm happy to take questions.
Great. Thank you, Filip. Really fantastic talk. We could probably have time for maybe just a question or so. Anybody have any questions? Ryan.
Yes, Ryan.
Filip, really nice talk. At the very end there, you mentioned something about engineering the ARA-C promoter or removing that. I didn't quite catch. You were basically saying you're making that constitutive, or I couldn't quite make sure.
Okay. Right
there, yeah.
So if you take the recoded strain and just remove the whole bad operon, which basically metabolizes arabinose,
right?
You remove ARA-C and you make ARA-E constitutively expressed by using this promoter from the Anderson library. You can, like if you look here, for example, at these bands where we have, for example, as proteins here, for example, we have TAF-B, which is elongation factor TU, and FAS-A, we get higher yields and faster protein production compared to just inducing this whole system with arabinose. So what I'm saying is, if you want to have your synthetase and you want to use your p-evol vector, where one of the synthetase is arabinose induced, to save you a lot of effort, just remove the bad operon, and you can, in these recoded strains, basically fine-tune gene expression by using arabinose, just by simply doing this trick.
That makes sense.
Have done, of course,
really,
more extensive
So really what you're
characterizations.
doing is you're making your cell line arabinose compatible for induction.
Absolutely, yes.
Okay, thank you.